The overall goal of this method is to establish a xenograft mouse model for mantle cell lymphoma to develop treatment strategies for this disease.
This method can help answer key questions in the field of hemato-oncology, such as therapeutic outcome of different treatment strategies. The main advantage of this technique is that it allows us to study the effect of drugs on human lymphoma cells in vivo. The implications of this technique extend towards therapy for mantle cell lymphoma because using this model, it is not only feasible but also convenient to study the effect of therapy on patient-derived cells. Though this method can provide insight into therapeutic aspects of mantle cell lymphoma, it can also be applied to study different types of lymphomas. Generally, individuals new to this method will struggle because it is difficult to obtain patient-derived xenografts.
Well, we first had the idea of this method when we wanted to study B-cell migration in vivo and the effects of some therapies on this process.
Dilute 12 milliliters of mantle cell lymphoma blood sample with equal volumes of RPMI 1640 medium. Then, invert the density gradient media several times before using. Aspirate 15 milliliters of density gradient media in a 15 milliliter centrifuge tube. With the help of an aspirator, carefully and gently add the diluted blood sample on top of the density gradient media without mixing.